Welcome to the Plant Cells Lab. In this lab, we'll be taking a closer look at the microscopic building blocks that make up plants. Even though we're working online, this lab is designed to mirror what you would experience in a face-to-face laboratory, observing specimens, making detailed drawings, and connecting structure to function. By the end of this lab, you should feel confident identifying major plant cell structures and explaining why they look the way they do. Before we begin working with the microscope images, let's take a moment to review the learning goals for this lab. By the end of this activity, you should be able to identify the major structures of a plant cell and explain what each of those structures does. This includes recognizing organelles such as the cell wall, nucleus, chloroplast, vacuole, and other plastids, and understanding how each contributes to the cell's function. You'll also explore osmosis, which is the movement of water across a membrane, and see how it affects plant cells in different environments. Understanding osmosis is essential because it explains how plants maintain structure, transport water, and respond to changes in their environment. Finally, you'll compare hypotonic, hypertonic, and isotonic conditions and observe how these different environments impact plant cells. These concepts will come up again throughout the course, so this lab helps build an important foundation. As you work through the lab, keep these objectives in mind. They will guide your observations, drawings, and explanations. Before we begin the Plant Cells Lab, I want to take a few minutes to introduce some key ideas that will help you be successful throughout this section. This short orientation will give you the background you need to understand what you're looking at when you observe plant cells under the microscope. Plants may look simple on the outside, but at the cellular level, they are highly organized and specialized. By studying plant cells, we can better understand how plants grow, store energy, transport materials, and respond to their environment. Throughout this lab series, you'll see how structure and function are closely connected. A cell is the smallest unit capable of carrying out all life processes. Whether it's a single-celled bacterium or a complex plant made of millions of cells, everything living is built from cells. In this course, we focus on understanding how different cell types support different functions. Cells fall into two broad categories, prokaryotic and eukaryotic. Prokaryotic cells, such as bacteria, do not have a nucleus or membrane-bound organelles. Plant cells, on the other hand, are eukaryotic. They have a nucleus and many specialized structures that allow them to carry out complex functions. Ribosomes are found in every living cell, whether prokaryotic or eukaryotic. They read messenger RNA or mRNA, which carries instructions copied from DNA. Using these instructions, ribosomes assemble amino acids into proteins, molecules that perform most of the work inside cells. This shared feature across all life forms highlights how fundamentally important protein synthesis is. One of the most important ideas in cell biology is the endosymbiotic theory proposed by Dr. Lynn Margulis in 1967. This theory suggests that long ago, larger ancestral cells engulfed smaller prokaryotic cells. Instead of being digested, these smaller cells formed a mutually beneficial relationship within the host cell. Over time, these symbiotic partners evolved into organelles, specifically mitochondria and chloroplasts. Several lines of evidence support the endosymbiotic theory. Mitochondria and chloroplasts contain their own DNA, which is circular like bacterial DNA. They also divide independently of the cell and are similar in size to many bacteria. These similarities strongly support the idea that these organelles originated as free-living organisms. As we move into observing actual plant cells, keep in mind that structure and function are always linked. Chloroplasts allow plants to capture light energy and produce sugars. Mitochondria convert that energy into a usable form for the cell. Throughout this lab, your goal is to recognize how cell structures support the specific needs of the plant. Before we start looking at plant cells under the microscope, it's important to understand how plant cells compare to animal cells. Both plant and animal cells are eukaryotic, which means they contain a nucleus and membrane-bound organelles. However, there are several key differences that affect how these cells function. Plant cells have a cell wall, which gives them structure and rigidity. This is something animal cells do not have. Plant cells also contain chloroplasts, which allow them to perform photosynthesis, something animals cannot do. Another major difference is the large central vacuole found in plant cells. This structure helps maintain cell shape, stores water and nutrients, and plays a role in pressure inside the cell. Animal cells, on the other hand, tend to have a more rounded or irregular shape and rely on different mechanisms for support and energy. As you move throughout this lab, keep these differences in mind. When you observe plant cells, think about how their structure supports their function and how that function differs from what you would see in animal cells. In this lab, you'll observe several different plant tissues, each with a unique structure and function. As you move through the activities, think about how the appearance of each cell type reflects its role in the plant, whether that's protection, storage, photosynthesis, or transport. Now that you have the background you need, we're ready to begin Activity 1, where you'll examine onion epidermal cells. Remember to pause when prompted, take your time with your observations, and focus on understanding what you're seeing, not just identifying parts. Play the next video for Activity 1.